Welcome. I'm David Schenk, Senior Advisor to Cure Alzheimer's Fund, a nonprofit dedicated to preventing Alzheimer's disease and ultimately stopping it as soon as we can. Today, we're lucky to have some time with Dr. Beth Stevens, a developmental neurobiologist at Boston Children's Hospital. She runs her own lab here, and she's agreed to tell us all about the important research she's doing. So, Dr. Stevens, thank you for joining us.
Thank you.
Thank you for being here.
Yeah,
thank you for
having me.
allowing us into your space and taking the time.
Absolutely.
So let's talk first about how you got into Alzheimer's. You did not start with Alzheimer's. How did that?
Yeah.
What were you doing before that, and how did you get here?
Right. So, I'm a developmental neurobiologist, and for as long as I can remember, we've been studying how the brain wires up and how connections between neurons, synapses form and get sculpted. And that's sort of a pretty fundamental process in brain development. And yet we know very little about the molecules and the mechanisms. So a lot of the effort early on, both as a postdoc at Stanford with Ben Barris and early days in my lab, was really just basic science, trying to understand how that worked. And it turned out that we unexpectedly uncovered a role for a group of immune molecules called complement and an immune cell called microglia in this process. And we became interested in the Alzheimer's angle because it turned out that we knew the genetics pointed to these cells, microglia. Genetics also pointed to complement. And we knew from lots of literature that these cells and this pathway was activated or aberrant in Alzheimer's disease, but no one ever really thought about it, how it was working. In fact, it was often thought to be sort of a secondary, downstream component of neuroinflammation, for example. And when we started really thinking about and learning about how this mechanism was working to eliminate synapses, it brought together another fundamental hallmark of Alzheimer's, which is the early loss of synapses. In fact, we know that that happens years before some of the pathological and clinical hallmarks of AD. And so we know synapses are vulnerable, but yet we don't know why. So we started putting these two ideas together and wondered, could the pathway we've been studying in the developing brain become aberrantly activated very early in Alzheimer's? And could it be driving or mediating the synapse loss? And if so, could we take what we've been learning from the developing brain and apply that to Alzheimer's?
Okay, so for the next half hour, I'm going to unpack what you just said, because there's so many interesting things there, and I want to make sure that the people watching, who are most of them are not scientists, really understand this stuff in the most approachable way. So let's talk about what synapses are. And I know there's neurons, which are brain cells, and synapses are really the connections between those. It's one nerve cell talking to another, right?
Right. So synapses are the connections or the junction of communication between neurons. It's how neurons talk to each other. And of course, a typical neuron can form up to 20,000 synaptic connections from other neurons. And ensembles of these connections form circuits in our brains. And that allows us to do all kinds of things. It actually regulates our behaviors, our thoughts, memories, for example.
Right.
And so what we know is that the synapses are necessary for this communication. And when synapses start to fall apart or become lost, it can impact, initially not a lot, but as enough synapses start to go, especially in important circuits involved in things like memory, that can be a big problem.
Right, so when you have a thought, or you have a memory, or you have a habit or something, it really is in that chemical electrical connection between this kind of network of neurons.
Right.
When you disrupt that, the thought and memory disappears.
Right.
Right.
And so because a neuron can form so many of these connections, a subtle loss of a few synapses may not be noticeable. But when a lot of synapses start to go, especially synapses that are really important for particular things like memory, you'll start to notice symptoms, right? And so during development, this loss of synapses is actually a good thing. It's part of this process called synaptic pruning, which is a mechanism by which our connections in our brain wire up properly. And it turns out that we start off early in development with an excess of synapses.
Meaning like when we're infants?
Yeah, when we're first, right after birth and up to the first many years, we have all these extra connections in the brain. And then through this normal developmental process called pruning, a large number of these extra connections get pruned away while others get strengthened and maintained. It's sort of the idea of use it or lose it. So connections between neurons that are meaningful to us get strengthened. That allows things like behavior or memory to form. Whereas those connections that are less important get pruned. And it's this way by which you get specific connections in the brain that allows precise actions. So this is normal. So losing thousands of synapses during development is normal and good. And that's what we've been studying much of my career, right? But it turns out that losing synapses in disease is not good because it's past that critical period, if you will.
At a certain point of age, let's say whatever, you tell me what the age is, you should no longer be pruning back your synapses, you should only be building them, essentially?
Yeah, so it turns out there's different critical periods of pruning, and it actually happens in different parts of the brain at different times. So parts of your brain, like sensory systems that control things like vision, tend to get pruned pretty early in development, where parts of your brain, like the frontal cortex, that are involved in executive function, those can continue pruning into early adolescence. So there's different windows in which this normally happens, but in general, this process of developmental pruning has this period and then it ends. And then for the most part, you'd not want to have pruning continuing on throughout life and losing all these synapses. So there could be some subtle remodeling that happens in parts of your brain, like the hippocampus, which is involved in memories and learning and memory. That's what we would call synaptic plasticity, where there's remodeling that might be going on during learning, for example. But it's not to the extent that it would happen during development. So it's a bit of a different process. What's interesting is that in almost many of the neurodegenerative diseases, ranging from Alzheimer's, frontal temporal dementia, Huntington's disease, glaucoma, in all of these diseases, although they're quite different with respect to the genetics, with the circuit or the part of the brain that's affected, they all involve this early loss or dysfunction of synapses. It seems to be a common hallmark. And that raises a very intriguing question that we've been very interested in, which is, could it be that in the end they're converging on a common pathway that could then drive the synapse loss? And if that were true, that would offer a lot of opportunities for therapeutic intervention.
Meaning at a certain point, it's the same disease at some point along the way.
It may converge and onto this synapse loss. And so this pathway that we've been studying, this immune-related pathway, which we can talk more about, seems to be relevant to a lot of these different diseases in addition to Alzheimer's disease. And so by learning from development or another disease, it can also shed insight into Alzheimer's.
Developmental neurobiologists have known about the pruning and how it's not supposed to go on. And what did you start to discover when you were working with Ben Barres, and what was the big insight there?
Right. So we were studying synapse formation and synapse development. And back then, when I joined Ben's lab, there was an unexpected result that came out of an unbiased screen, if you will. So something where you didn't know what you were going to find. And it was basically the discovery that these molecules called complement, which are traditionally associated with the innate immune system, that this particular group of molecules was very much upregulated in neurons that are undergoing pruning, right? So this was like a surprise. It was like, well, first of all, what are these molecules doing in the developing healthy brain? What are they doing expressed in neurons? And there's no infection, there's no inflammation, this is normal. And that led us to start to think about like understanding it more deeply. And for this, this was at a time, over almost 15 years ago now, where the idea of immune molecules in the brain was still a pretty new concept. We didn't really think about it that way. But it turns out that there's a whole bunch of these molecules that are really traditionally associated with the immune system that are normally working in the brain that we've largely ignored until fairly recently. So this opened up a whole new area. And there's many labs that are continually discovering other examples of this, but complement was one. And that complement discovery is what quickly led us into this pruning idea. And that is because when you looked in the immunology textbooks at what complement traditionally does, its main role or its main job is to tag unwanted cells or bacterial cells for rapid removal. In other words, elimination of a pathogen. That's how it works in the immune system. These are a group of secreted molecules that literally bind, like tag or coat something like cream cheese on a bagel, just like coat it. And when those molecules do that, it recruits immune cells called macrophages to it, and then those are like Pac-Man, and they literally will recognize the coating and eat it. So complement's an eat me signal, essentially. So we started thinking, well, maybe because its main job in the immune system is to clear and to remove things, maybe its job in the developing brain is to help remove synapses. And lo and behold, when we looked with the right tools to investigate where complement was, we found it was tagging synapses in the developing mouse brain. And this discovery, was initially unexpected, but it kind of then started to make sense because we knew a lot about this group of molecules in the immune system. So it was a good example of learning from what the immunologists have been studying and then applying it to the brain.
Okay.
So from there, we're like, okay, complement's involved in this process, and that's what led us to the microglia, the immune cells. So in the brain, there's the resident immune cells, the sort of Pac-Man, are called microglia. And indeed, one of the first set of studies we did in my lab was to ask whether the microglia in our brains were gobbling up synapses. And we discovered indeed they were. And so this was the first evidence that microglia...
That was not known until...
No.
Wow, okay.
So that's opened up a whole nother area in my lab, and many other labs now are starting to think about the fact that these cells, microglia, are incredibly interesting and complex, and they have far more functions than we first knew about. And much of what we knew about microglia up until that time was in the context of injury or disease, where we knew, for example, they were major mediators of inflammation. They actually release a lot of the signals that cause neuroinflammation. And we also knew in the context of Alzheimer's disease that because they're like the Pac-Man, one of their good jobs in the brain is to gobble up amyloid plaques, for example,
right,
or dead cells. So that's a good role of microglia and what a lot of the field at that time had focused on. But the idea that they could have completely other roles, normal day jobs, if you will, that involve things like synapse development and remodeling, like that was sort of a new idea at the time. And so for many years in the lab, we've been just digging in on trying to understand like how that was even working. And the idea of that if we could understand how they recognize synapses and how that process worked, that could shed new light into how these cells go awry in Alzheimer's disease. And that's been the approach we've been taking.
So microglia are it's part of the immune system, right? And they're responding to injury, right? What do we, before discovering that they are eating these synapses, what did we think they were doing in the brain, or were they not known to be in the brain? What was?
So we knew they could eat things like plaques or dead cells,
okay,
right? So that the role of a phagocyte was established. In fact, there are probably our main phagocytic cells in the brain. Now we know also the astrocytes can also do that, but the microglia were known to be like sort of our professional phagocytes, called into action when needed, right? But during development, like no one had ever really thought about them. In fact, as a developmental neurobiologist, I completely ignore them because they're the only cell not born in the brain, and they actually come into the brain after birth. And so they were kind of like thought of as even as a glial biologist. I ignored these cells because they just didn't fit with the timing of when all this was happening. But what we now know with more modern tools and technology is that these cells actually come into the brain really early in development. In fact, before birth. And in fact, they're the first glial cell in the brain, the first non-neuronal cell in the brain. And now we know that they have probably all kinds of roles in development that we never appreciated before. But the thing that probably really got my attention is that we can, and other researchers have looked into the brains of a mouse, where you can use strategies where these microglia can be labeled with a fluorescent protein. And imaging studies allowed one to peer into a mouse brain and watch these cells.
Alive
cells. You can make a little window into a mouse's brain, and because these cells were bright green, you could peer in and just watch them and say, what are they doing? And so this was work that was done many years ago now by Axel Nimmeron and another pioneer from Wenbiao Gan's lab. These two groups were the very first to ever image microglia in real time. And what we realized from those pioneering studies is that one of the things microglia are doing all the time, probably right now as you and I are speaking, is that they're moving their processes and literally surveying the brain all the time. It's like what they do. They're essentially surveying. As their immune cells, it actually makes a lot of sense. That's their job.
Right.
But no other cell in the brain can do this, can move their processes so dynamically. And so even though they make up only 10% of the cells of the brain, because they're so dynamic and they can survey, it's estimated that they can survey the entire brain in a very short amount of time because they can move.
You make them sound like they're independent little robots inside our head that are doing their own
I wish
thing.
I had a movie to show you, but they're so incredibly interesting.
Right.
And so one of the things we then realized what they were surveying, because probably many things are surveying, but when you overlay synapses, which we can also label with, let's say, a different color fluorescent protein, red,
Right.
when we put neurons and microglia together with two colors, then we realize that one of the things they're doing is literally surveying and touching synapses all the time. They're essentially sensing what's going on with the synapse in a very interesting way. And that is what led us to wonder, well, when they're touching a synapse, could they actually be nibbling one off? Could they be actually engulfing synapses? So we designed a set of experiments in the mouse using these kinds of fluorescent tools to watch them do this, and indeed discovered that they're literally going in and plucking off bits of synapses. And again, during development, this is normal, good. This was that discovery.
So does that mean they're a part of the pruning process?
Yeah, they're the pruners.
They're the pruners.
They're the pruners. So they're literally like professional pruners.
Wow.
And of course, when things go wrong, if you were to literally injure the brain, and that's been done, then they can get really kind of angry, and then they can chomp off much more than a synapse.
Right.
But the discovery that they were very selective with the synapses they were eating was a new discovery in the sense that they were part of this pruning process in ways that we didn't appreciate before.
Okay.
And then that raised the question, well, how does a microglia know which synapse to prune and which ones to leave alone? Because only a subset of these synapses get pruned. Remember the idea of use it or lose it. And that's where complement comes in. Because complement, we found, is selectively targeting subsets of synapses, and they're one of the recognition signals. They're the eat me signal. So if microglia are surveying along and they see complement on that synapse but not that synapse, that would then direct them over to that synapse. And we indeed showed that that was the case. There's probably many other ways this works. In fact, we know there are probably a lot of other molecules besides complement, but this was one way of putting those two ideas together. It's a way of basically telling the microglia which synapse to eat. So now, what does this have to do with Alzheimer's disease, right? So this is all cool in development and neat, but you want to understand how this relates to Alzheimer's. Now, I told you at the beginning that Alzheimer's involves a very early loss of synapses. And another interesting and important aspect of this is that synapses in Alzheimer's they're not lost everywhere. There's actually vulnerable brain regions and vulnerable circuits that happen in Alzheimer's that include areas like the hippocampus or frontal cortex, which is involved in that executive function. So we decided to ask, in Alzheimer's disease, could the microglia be aberrantly eating these synapses very early? Could they be part of the process? So we used some of the same tools I just told you about and did these kind of synaptic eating experiments in mouse models of Alzheimer's, where we could label the microglia with one color, label the synapses with another, survey the brain and ask, okay, in areas where we know synapse loss happens, are the microglia part of that process? And we designed and did a number of different experiments to show that yes, they do engulf synapses, and they're getting called into action quite early in the process. And more importantly, if we block their ability to do this, so if we block the receptor on them so they can't see the signal anymore, or if we block complement itself, microglia don't do this anymore. And when we block that, it's protecting the synapses. And in a more recent study, even protecting some of the memory loss and cognitive impairment.
So there's a healthy version of them pruning the synapses,
right?
And then there's an unhealthy version, and the unhealthy can lead to Alzheimer's, and it sounds like other diseases.
Okay. Yes.
And you've shown all that. Do you know what separates the healthy from what tips them over?
Yeah.
What is causing the microglia to
That's the $10 million question.
Okay.
That is what we still need to figure out. So if we could figure out what activates them, what triggers this whole thing, what are the signals that make them suddenly become from good to bad, that would be huge. And we're working on that very hard. In fact, we're working on that back in our normal developmental context because we have some clues about what does that. And we think some of those clues might also be called back into action in Alzheimer's disease. So we don't have an answer to that question yet, but you've just touched on a very important area of investigation in my lab is to try to figure out what's regulating this process. There are positive and negative regulators, just gas and brakes. We want to figure out what those are. And if we could do that, that potentially has therapeutic implications. If we could understand really how to sort of switch this thing from a good thing to a bad thing to a good thing.
So potentially, if you can find that point where they switch over from being these beneficent pruners to these monstrous eaters, you could stop. You could keep them from becoming these monsters.
Right. And that brings us to really even a broader question beyond complement and what we're studying in the lab, which is, in the last maybe 5 years or so, there's now a sort of a spotlight on these cells. And the reason for the spotlight, even though we've known for decades that microglia become activated or angry in Alzheimer's disease and other diseases as well, they sort of respond to injury in the ways I just told you. But what's really been game-changing in the Alzheimer's field is that human genetics, that are emerging now are continually implicating these cells as not just a downstream player, but as a causal player. And that is that many of the genes that have been implicated and discovered to play a role in Alzheimer's risk, to increased risk, are either expressed exclusively in those cells or enriched in those cells compared to other cells.
Wow.
And that tells us that these cells are more important than ever to understand how and when they become dysfunctional, because we still don't really understand that. And so there's many different ways that they could be increasing risk. They could be not eating plaques anymore, and there's a lot of research devoted to that aspect. Meaning if they can't engulf and gobble up plaques, that would be a bad thing. You'd want to clear those plaques. So that's one mechanism. The other is they could be contributing to too much inflammation because of these different genetic risk factors. That's another angle that the field's focused on. If you could try to prevent them from releasing things that cause inflammation, that might be a good way to kind of push them back. The other that we're focusing on is the synaptic angle. If we can figure out how to make them not eat synapses but still eat plaques. So this is a balancing act here. And that raises probably the most important question that the field, in my opinion, needs to figure out, which is understanding better what microglia are doing in the brain normally and how they're switching states from this good guy to the bad guy. Because if we could figure that out, then we can start to target our therapeutic approaches and our mechanisms to try to get the good microglia to be enhanced and the bad microglia to be suppressed. But we don't yet know how to do that.
Right. So now let me kind of try to marry this with something we talk about all the time in programs like this, which is we talk about amyloid all the time, and we talk about how we've got this hypothesis, which everything seems to have so far that I know of. Just the more data that comes in, the more that proves this hypothesis, and Rudy talks about it all the time, which is that, to put it really simply, starting in our 20s or 30s or 40s, we have this amyloid, which is doing this healthy stuff in our brain, having to do with the innate immune system, but then we start to develop too much of it. It starts to build up in our brains, and then that. And we often talk about that as being the kind of kickoff process. That then kicks off. That starts the production of tau, the tau proteins, and also the inflammation. So how does what you're talking about with the pruning and with because I understand. So let me finish the question, then I'll ask a follow-up. That I understand that you have actually detected that some of the synaptic stuff that's going bad we're detecting that before we see the plaques in the brain. So how can that be if we're also thinking that the amyloid happens decades and decades before the disease?
Because there's a form of amyloid before it makes these fibrillar plaques that are quite toxic to neurons and synapses, and that can exist quite early before the plaques are actually formed. Now, in addition to the fact that once the plaques form, that can also cause synapse loss. So basically, plaques and amyloid are bad for synapses when it gets dysregulated this way. But what we did, and we wanted to ask the question, could microglia and this pruning be happening independent of plaques? So what we did is we looked at an animal model so early before any plaques were there. And we simply asked, does synapse loss happen, at least in these animal models of Alzheimer's, at that early stage? How early is the synapse loss happening? We found it was actually quite early, only a couple months in, which most people weren't looking that early. And there were no plaques in sight when this pruning process started.
A couple months into what?
Into the course of the disease for an animal model.
Oh, okay, all right.
So normally plaques would start to form in a lot of these mouse models at like 4 months, 5 months, and more. We were rewinding the clock back to 2 months, 3 months, and could see the synapse loss, no plaques yet, and then eventually the plaques, of course, start to form. So we decided to say, well, we know that the plaques are really important and very detrimental, but let's just focus on this little window here, because in a mouse you can control it, in people you really can't. And that's what we did in this study that was published last year, is to just get at this question of, could this aberrant pruning process happen independent of plaques? And we showed, at least in animal models, that it did.
Meaning also independently of the amyloid before it even forms as plaques?
But the amyloid was there, and
Okay.
it was actually driving the synapse loss, as had been
okay.
published before. So, but they weren't plaques. It was more the secreted form, this oligomeric form, before it makes plaques.
Okay.
And that is what Dennis Selko and Bernardo Sabatini and many others have now shown, that form of amyloid beta is toxic to neurons and synapses, even in its soluble form.
Right,
so if
right.
you
Okay.
put A beta, the soluble form, onto a culture of neurons in synapses will start to go. And we could see that also in the animal models as well. So this is quite an early event, and it is amyloid, absolutely, but it's not quite the plaques yet.
Right. So in the chicken or egg thing, that just going and putting it really simply, where we talk about the amyloid being first, you're not contradicting that. You're
No.
saying the soluble beta amyloid is there first, and that getting out of control number-wise,
Yes.
the accumulation of that still looks like the very first event in the earliest part, the very, very pre-symptomatic earliest part of Alzheimer's.
It's quite early, right.
And but then pretty early on, that is leading to this synaptic event, which is also very early part of the process, way before we see symptoms in the
exactly.
disease.
But I should also say, to put this together with what else is known, that those plaques, and in people, we don't know exactly what the window is. It could be that the plaques are forming sooner than this, but at least in animal models, I can tell you. But once those plaques start to form, they release these soluble synaptotoxic forms of A beta, and there's been really, really nice evidence that shows synapse loss also happens when plaques are there. So synapse loss can happen in both cases, but the very earliest form of synapse loss that we were studying didn't have plaques there. But it just gets at this point of, we want to go as early as possible, and we want to try to save the synapse.
Right.
And the reason for that, of course, is that synapse loss is a stronger correlate of cognitive decline and memory impairment than having plaques or no plaques. In other words, there are people that have lots of plaques in their brains, but they're cognitively okay.
Right.
Why is that?
Right.
And what we did in a subsequent follow-up study that was published this year, and this was a study led by Cynthia Lemire, who's here at Harvard, and we've been collaborating for a number of years. Cindy decided to look in these animal models where you can remove complement, C3. This is this complement protein. Basically make a mouse model where it doesn't have that eat me signal. And basically ask the question, if we save the synapse, do we also protect against all of the other pathological hallmarks as well as memory impairment and cognitive impairment in these animals? And what that study showed is that protecting the synapse by removing this signal also led to a protection in some of the cognitive and memory-related behaviors. However, there were more plaques in these animals. Now that's an important point, but it's completely consistent with the role of complement in helping to get plaques cleared.
So we've gotten a lot of detail here. I'm sure people are a
too
little
much.
numb, but
Yes.
but it's still, I mean, it's fascinating, it's important, and that's Alzheimer's is very complicated. Let me ask you, before we try to wrap it up, let me just ask you, I'm sure a lot of people are wondering out there that they've heard so much about tau, the other kind of aberrant protein that's been famous, and there have been lots of studies for years and years showing it. That actually correlates really well to the disease process. So is there any connection there that you're seeing, or is that not really turning up in your
We are very interested in knowing the answer of whether this same mechanism I've been talking about with microglia and complement is also involved in the tauopathies.
Okay.
And so there are people looking at that. We don't know the answer. My suspicion would be it's all somehow part of the same bigger pathway.
Right.
But we don't know yet.
But it looks like, if I'm getting your timeline correct, it looks like it would be the beta amyloid first, then the synaptic loss process going awry, and then the tau. That would be kind of the
More than
sequence.
likely, right. But it needs to be formally tested in terms of what comes first. It's always this chicken and the egg question.
Right.
But an important one.
So we haven't really said exactly how Cure Alzheimer's Fund fits into your research and what they've allowed you to do that you wouldn't have been able to do otherwise. Can you speak to that?
Absolutely. So everything I've talked to you about today has been in animal models of Alzheimer's, which for us is the first step because it allows us to really tease out and test hypotheses, tease out mechanisms. But in the end, animals are not people, and we want to be able to try to translate what we've learned from these models towards patients and therapies. We want to understand if what we've been studying in a mouse is relevant to what's happening in human Alzheimer's brains. So what we are doing now, thanks to the support of Cure Alzheimer's Fund, is start to collaborate and use to test this hypothesis in human brain samples. So we can get autopsy brain tissue where we can look and see whether this complement and synapse loss and microglia are doing what they were doing in the mouse in the human brain. And the other angle is to look in cerebral spinal fluid, or CSF, to ask whether this pathway, which we believe happens quite early, could be increased in the CSF of patients with Alzheimer's. And if so, when does that happen? So there's already been collections of CSF over multiple different stages of disease, even at the preclinical stage. And there's been a lot of evidence that A beta, tau, even TREM2, another molecule that's been recently shown to be increased in CSF. We know these things change, and now we want to know how complement, or whether complement, is also changing, and how that fits with the other biomarkers that have been identified. So this is all ongoing right now, but we can now start to measure different components of these complement proteins in CSF of patients and ask whether there's changes, and if so, if it's happening early. And this might be a proxy for what's happening in the brain, right? So could it be telling us when our synapses are starting to go by seeing changes in these proteins coming out into the cerebral spinal fluid? So this is all work in progress, but it's exciting because now we can start to compare what we've learned from the mouse and start to look in human Alzheimer's.
That's all the time we have today. Thanks to Dr. Beth Stevens for spending so much time with us. Please tune in again. If you want to learn more about Cure Alzheimer's Fund, you can always go to curealz.org and ask us any question you want.